Good day, and thank you all for participating in today's webinar. My name is Terry Albarano, and I'm the Senior Marketing Manager for Omnicell's IV Solutions portfolio. We're excited to support this continuing education webinar, Beyond Use Date Strategies for Compounded Sterile Preparations, with Kevin Hansen, Assistant Director of Pharmacy at Moses H. Cone Memorial Hospital, and Brian Kelly with ARL Biopharma. I'd like to first cover a few housekeeping items for today. There will be an opportunity at the end of the presentation for a Q&A session with both speakers, both Brian and Kevin. And what you need to do is submit your questions in the Ask a Question box on your screen. In addition, if you experience any tech-related issues, there's a question mark icon in the upper right-hand corner of your screen, and you can submit any of those issues via that icon. As a requirement for ACPE accreditation, we need you to complete an online evaluation. This evaluation is of your perception of the continuing education course. We need your comments, criticisms, and suggestions on this program. At the end of the webinar, you will be provided this information. So at the end of the webinar, we'll take you directly to the survey you need to complete for your continuing education credit. Post-webinar, we will also provide attendees the QR code and URL that you could also use to complete this survey. There's also a downloadable version of today's presentation slides available on the webinar platform on the event resources page. So feel free to download this presentation. And I'm going to go ahead and kick off the presentation now that we've covered some of those housekeeping items. Again, the title of the webinar is Beyond Use Date Strategies for Compounded Sterile Preparations. Our speakers are Kevin N. Hansen, Assistant Director of Pharmacy at Moses H. Cone Memorial Hospital. And we also have Brian Kelly, Director of Business Development with ARL Biopharma. And I'd like to just review a few disclosures today. Neither of our speakers declare any conflicts of interest, real or apparent, and no financial interest in any company, product, or service mentioned in this program, including grants, employment, gifts, stock holdings, and honoraria. And the objectives for today's continuing educational program are to review the USP 797 revisions impacting beyond use date assignment for compounded sterile preparations, outline additional testing methods and USP compounded monographs for sterile injectables, differentiate sterility testing methods for compounded sterile preparations, and apply strategies to extend beyond use dates for compounded sterile preparations. There's also a list of the abbreviations that will be used in this presentation, and I'm going to kick the presentation off now to Dr. Hansen, who will be covering Objective 1. Kevin?
Great. Thank you so much, Terry, and thanks to Omnicell for hosting this presentation webinar today, and thanks for all of those dialed in to hear the conversation, and hopefully at the end have an opportunity to participate in some question and answers. So I'll go ahead and kick us off here with our first objective of reviewing these USP 797 revisions, specifically to that of the beyond use date assignment for compounded sterile preparations. There's certainly been a number of questions around this topic, and so felt it was very pertinent to put some material together talking about the changes and then also some strategies of how to work through some of these revisions. And so I think it's pertinent to go ahead and start with, differentiating and going back to the source of differentiating between the expiration date and a beyond use date to really put this in context. And so, of course, we know the expiration dates. We see these are assigned by the manufacturers. We see these on these commercially manufactured products, APIs, excipients. And these are a date that's, that the product should not be used because it no longer meets those, the requirements listed in that compendial monograph, or may not be potent any longer. And we know that it's based on extensive analytical and performance testing of these products. Their sterility, chemical and physical stability, package integrity, so on and so forth. So it's a very scientific, applied a date to that product. Now, differentiating that to a beyond use date, this is different as this is assigned by the compounder. And so, of course, for our compounded sterile products or non-sterile preparations, and it's different than an expiration date that is the time of which it should not be administered. So this date does stop at that time of where that administration begins and where Chapter 797 no longer applies. And it's based on conservative estimates and more risk-based strategies for the period of which that preparation is expected to retain those characteristic properties. And so, of course, we do use scientific information and best practices for sterile compounding to obtain this beyond use date, and is based off of stability factors and sterility factors. So looking at some of the elements that goes into each of those buckets, so on stability factors, we know the chemical properties of the compound, the physical properties of that compound, and then also understanding how the compatibility of that admixture relates to that container closure system, and can it maintain the sterility and the stability of that while it's in that container over the beyond use date of that product. And then, of course, where 797 has a lot of focus on is those sterility factors. So now focusing on the environment in which the CSP is being prepared, whether that's in your segregated compounding area or that's in your true cleanroom suite. And now we have some differentiation there. Whether or not you're using aseptic preparation, so sterile to sterile, or some type of sterile filtration, or true, sterilization methods or terminal sterilization. The components, whether you're using sterile or non-sterile, whether or not you're performing a sterility test, and then of course your storage conditions and the packaging and the temperature that you're storing those in from that standpoint. So we are all very familiar with the, we'll now call this the old, I guess, or the previous or the current, I guess you could say, 797 beyond use date table. And we know that it's based off of the risk category. And so there's a number of elements that goes into determining that, the number of products you have, the complexity of those admixtures, and as we look at this table, we have the risk category, and then we have the temperature of that storage of that final CSP. And then we're provided either in hours or in days what that maximum allowable beyond use date can be applied. However, in the previous setting, in the 797, it listed in there that these are dates in the absence of passing a sterility test, that USP 71 sterility test, they can't exceed these dates. And so then that would there imply that if you did a sterility test, that you were allowed to exceed those dates. And we'll come back to this concept here in a moment. Now, contrasting and comparing that to the revised USP 797, the 2019 version that's going to go into effective December 1 of this year, and we'll look just at the Category 2 CSPs, so these are ones that are prepared within a cleanroom suite. We can see that there's been some differences of now what's included. So first and foremost, the differentiation between Category 1 and Category 2 of being where they are compounded. So that's taking into account that there's differences of sterility or aseptic control based off of the environment that you're compounding these products in. And so for those Category 2 CSPs, we're allowed to apply these longer dates compared to the Category 1s. Well, it also now includes some additional information in here that we need to consider when we're applying these beyond use dates. So one is the method. Are we aseptically processing these CSPs, so that's sterile to sterile transfers or sterile filtration, or are we terminally sterilizing these end products? So are we doing gamma irradiation or some type of steam or heat, if you will, from an autoclave? The next category is whether or not you're doing a sterility test. So you can see if you're not performing that USP 71 sterility test and also having a passing result, the table now gives some information about what dates are allowed. And the next category is the ingredients. So looking at are your starting ingredients sterile? Do you have any non-sterile? And then there's some differentiation on the table here. And the last element that the table provides is the storage conditions that we're all familiar with, whether or not you're going to store it at room temperature, refrigerated, or frozen conditions. So the one difference between this and the previous table is that phrase, in the absence of sterility test, is no longer included, because you can see that now sterility test is added into this table, and now we are defined a maximum value of what we can assign from a beyond use date perspective. So when we put both of these side by side where we can compare and contrast, the first question to ask is, does this now allow for extended beyond use dates greater than those listed in the beyond use date table? In the old version, the 2008 version, the answer was yes, you could do that. In the new version, the answer is no, you can't. You have to follow the table exactly of what's in there to get those dates that are there, and we'll talk about one exception to that rule later in this presentation. Methods for longer beyond use dates. And so in the previous chapter, it included your storage condition, your refrigeration or room temperature, your starting ingredients, whether you're doing the USP 797 defined high-risk compounding of using non-sterile ingredients that had a shortened beyond use date allowance to that, and then whether or not you were passing a sterility test. So the new version still has that, however, they do add into that now the sterilization method. In the old chapter, a maximum beyond use date allowed was not specified. However, using this new table in the revised 797, you can see that the longest possible date that you can assign within this table is 90 days. And that is, to achieve that 90 days, you would have to terminally sterilize your product, it would have to pass a USP 71 sterility test, and it would need to be stored at frozen conditions. So you can see that based off of the changes here, there's been some significant changes, and would need to evaluate our practices to ensure that we're going to be compliant with this. So during the second revision of 797, early on in I think it was about in the table of contents, there was a content in here that the expert committee made a comment that they're considering development of a resource, and the resource would include some additional information to compounders about what are those additional testing and what is the criteria to use those testing to properly extend a beyond use date. So it really goes beyond just having a USP 71 sterility test. There's stability indicating method assays with forced degradation. You have your endotoxin test, container closure integrity, particulate matter, and the list goes on. And so it'd be very interesting to see when and if this resource gets released, and we'll be talking through some of those additional tests. Brian will be discussing those later on in this presentation. So the one caveat or exception to the rule within that table that I wanted to bring out, so USP has clarified this in a frequently asked questions document on their website. If you have not visited that site, I have a link down here, or is certainly easy to find on the USP website. Highly encourage you to take a look at these frequently asked questions, because it can really help provide some context and some additional clarity on some of these concepts. So one of the frequently asked questions they have is about, can the beyond use date of a Category 2 CSP be extended beyond those listed in the table 11? And that was that beyond use date table we looked at a few slides earlier. And they state that if there is a compounded preparation monograph, again, these are USP compounded preparation monographs, for a specific CSP formulation, that beyond use date may be assigned if that CSP is prepared exactly according to that monograph. And so there's a section on additional tests that we'll go through and looking at the assay and making sure that we're within that criteria range. And then also the starting ingredients, everything has to be exact. And you do have an allowance now to quote unquote exceed or extend those dates beyond what's listed in the table there. So I think that becomes really important in our conversation, and also identifies a gap of we need more of these USP compounded monographs really in the future, and should be a focused target area for us. So I'm going to go ahead and turn it over to Brian now, who's going to talk about outlining some of these additional testing methods that compounders can use.
Okay, yeah, thank you, Kevin. So as Kevin mentioned, there are some compounded preparation monographs that will be a valuable tool for compounders because they do include the extended BUDs. So we're going to take a look at a couple of those monographs and the BUDs, and then go through the requirements of the monograph, because as Kevin just said, to be able to utilize the BUDs in those monographs, you must follow the monograph as a whole. Okay, so the first monograph we'll take a look at is sodium bicarb compounded injection. Okay, so whenever looking at any monograph, you always want to start with the definition. The definition provides a lot of valuable information, so don't skip that part. The eyes tend to go right to the table, but you want to make sure you're reading the definition there. You'll see in this definition that the acceptance criteria for the potency test is 95 to 105, so different than the standard 90 to 110. It also states that the preparation won't contain any preservatives. Has some information there about how to prepare the compound preparation and filtration and things. So make sure you're going through each of these line by line. A bit of a disclosure here, we are not showing the entire monograph, just so we hit on the key points that are relevant to this presentation. So these are the list of the tests that are in the monograph. The assay, or commonly referred to as potency. Notice that they have the requirement for the potency both on the sodium and the bicarbonate. That's to make sure that you're using the correct source of sodium and bicarbonate and didn't accidentally use sodium chloride, and if you only tested for the sodium, you wouldn't pick up on that. Also has pH with an acceptance criteria there. Sterility and endotoxin, again, a limit provided for you on the endotoxin test, and the particulate matter. There's a lot of focus in the new 797 about the importance of particulate matter. It talks in one part about, in the sterilization section, about potentially doing a pre-filtration with a 1.2 micron filter to reduce particulate matters prior to using your 0.22 micron filter for sterilization. So a lot of focus on particulate matter. The particulate matter is in all of these injected monographs. It's an important test, so and make sure you familiarize yourself with that test. Looking further down the monograph there, it has packaging and storage instructions, controlled room temperature, and then notice the beyond use date. So it's pretty clear here, if you do not complete the sterility and endotoxin test, then you have to use the beyond use dates in the table 11 that Kevin went through earlier for when the sterility and endotoxin tests are not completed. But if you have successfully completed the sterility and endotoxin test and followed the monograph exactly, then there is a beyond use date of 120 days. So so look for these monographs. The way you can find them is by either searching for the drug name or by searching USPNF for the word compounded. We'll pull up all of the monographs. So, okay, we'll take a look at another one. This one was an API or powder monograph. This next one here, the fentanyl citrate ropivacaine, is a sterile transfer. Again, so always start with the definition. This one is a 90 to 110. That's pretty standard, but I do want to point out that the 90 to 110 acceptance criteria for the potency on the fentanyl is based on the fentanyl free base. So even though you're using fentanyl citrate, a salt, to make the compound, the acceptance criteria is on fentanyl, the free base. That's shows up in the table there below too, where it says that you're using the fentanyl citrate injection, but you're transferring 200 micrograms of fentanyl. So make sure your calculations and things are adjusting for that. And then notice also below the table there, it has specifics on the manufacturer of the commercial products that were used in this sterile transfer, and then has some instructions on how to compound the preparation, and then a note about filtration. So so make sure you're paying attention to all the details. Again, it has the assay. There's two APIs there, so you can test the potency of both of the APIs. This one in particular has a purity test to make sure that you have the correct structure of ropivacaine. Again, the pH with a acceptable range there, sterility, endotoxin, and particulate matter testing again. So this one, if you follow the monograph as a whole, has a beyond use date of 90 days. So look for these compound preparation monographs, and there may be some beyond use dates that will be useful to you. Second thing we want to talk about here is sterility testing. So with so much emphasis on the sterility, on the BUDs, on whether or not sterility testing has been performed, this is going to be an important test because it's really going to be the batch release rate limiting test. What I mean by that is this test is going to be typically the longest test where you're waiting on its test results before you can release your batch, and that test will be eating into those use periods. So a lot of emphasis on this test. The USP 71 test is a 14-day test at a minimum. The samples have to incubate for 14 full days. So when setting up your testing programs and things, look for every opportunity to minimize any testing delays. Work with your laboratory and make sure you're getting the method suitability established ahead of time if you can. Give them a heads up of samples coming in. Anything you can do to get the method suit squared away ahead of time. Ship samples out the same day you make them. Use overnight shipping. Make sure the laboratory is starting the testing on the day of arrival. Again, look for every opportunity you can. So if you think about, unless you are able to basically hand the sample to the laboratory staff, and so they can get testing started the day of arrival, it's a 15-day process if you're going to be shipping them overnight at a minimum. So with a 15-day process, if it's an aseptically processed sample with sterility testing, you're down to 15 days at room temperature, 30 at refrigerated. So anything that any delays that you run into along the way is going to be eating into that time. So look for those and try to keep those to a minimum. I did want to hit on early release here. I know there's going to be some scenarios in which you can't afford to wait for a sterility test, but you still want to have the sterility testing conducted. So if you're going to, if you need to do an early release, USP does allow for that to happen. Of course, you would have to use the no sterility testing BUD, because the Table 11 says that for the longer sterility testing BUDs, the test has to be performed and passed, so it has to be completed. So if you're releasing before the completion of a sterility test, you have to use the shorter BUDs. But there is an allowance to do so, but if you do so, you must have procedures in place to notify prescribers and to determine if a recall is necessary. And then if you have, if a recall is necessary, you must have an SOP in place to dictate how that recall occurs. And note there at the bottom that the recall must include a procedure to notify appropriate regulatory bodies based on jurisdiction. Take a little time here to look into the USP 71 sampling, particularly because USP 797 did something outside of 71 when it came to batch sampling, so I wanted to hit on that. So USP 71 batch sizes greater than 40 units, you must sample according to Table 3. You have to round up. We're going to hit on that quite a bit, so make sure people understand that concept. The difference here for 797 is that it has an allowance to make less product but still be able to get a sterility test. So the first example there is a batch size of 1. If you have, just make 1, it says to make 10% of 1 rounded up, and so that would be make 1 extra. So 10% of 1.1 rounded up to the next whole would be 1 extra. So with the traditional 71 requirements, if you were just going to make 1, you would need to make 4 additional samples. So there is an allowance here for less samples to be tested, but it's not, if you send in 1, that would not be in alignment with 71, so it still wouldn't be USP 71 sampling, but it would be in the spirit of 797. You also want to make sure that the laboratory is sampling per table 2. We'll hit on more on that later. So USP Table 3, this is just a portion of the table, so we can look at it easy here on the presentation. But so make sure you have a reference to USP for the current table, and have this available. Have Table 3 available for your staff that is responsible for sampling batches for sterility testing. So I'm going to take a look at a couple examples here and work through the calculations. So in this first one, it's a pharmacy makes 45 10 ml syringes of neostigmine. You start on the left side of the table, work your way down the table until you find the appropriate line that applies to the type of preparation, the volume of the preparation, how many of the items are in the batch. So in this one, it's neostigmine's parenteral preparation, not more than 100 containers since you make 45. So then here's where you need to do the calculations, because the acceptance criteria is 10% or 4, whichever is greater. So with the batch size of 45, 10%, 4.5, rounded up is 5 syringes. So back to the table then, 5 syringes, which is 10%, or 4, whichever is greater. So you would need to submit 5 syringes for the sterility test. Same product here, but now the batch size has increased to 375 syringes. And so you're no longer, does the not more than 100 containers apply, but you now fall on the more than 100 but not more than 500. So 10 syringes need to be tested. No math there, just a straight 10. I mentioned earlier that the volume's important, so these are 1 liter bags of oxytocin. So it's still a parenteral preparation, but it has now fallen into a large volume parenteral. The USP Chapter 1 defines large volume parenterals as container closure systems that contain labeled as and contain greater than 100 mils. So that's key that you understand that's labeled as and contain greater than 100 mils to be considered a large volume parenteral, which is important because then you can, it changes the sampling requirements here. So notice it's now 2% or 10, whichever is less. So again, do the calculations, round up, reference back to the table. In this case, it'd be 4 bags. 4 bags is less than 10, so 4 bags need to be tested per USP 71. Mentioned earlier about Table 2. Again, this is just a portion of the table, but wanted to hit on this so that people understand that there's 2 portions of the sampling. One is how to sample the batch. The second portion is how to sample those samples that were selected out of the batch. Okay, so in this example, it's 5 10 mil vials. So you would look down the table and you see that it's 10 mil vials, so it falls in between 1 and 40. So that's half the contents of each container. But if you look at the title of that column there, it says for each medium. So if you have to take half the contents for each medium, that means all the contents of all of those 5 10 mil vials are going to be used in the sterility test. So I know there's some practices out there of just taking a portion of the vial, maybe putting it on a petri dish, or using some of these. There's some commercially available membrane filtration testing devices that only test a mil or 2. In neither one of those scenarios is in the spirit of a USP 71. So those are not USP 71 tests. So be careful with those. On the in USP 71, it does state that membrane filtration is the method of choice. There's a reason for that, and that's because it's the least likely methodology that contamination will occur while the sterility test is being conducted. But not all drug products can be filtered, so the preferred alternative method for those, like suspensions, is direct inoculation. Okay. but really with the kind of message we're trying to deliver in this webinar, what's important here is that is alternative methods, rapid sterility methods. So USP 797 does allow for the use of rapid sterility test methods, but the methods have to be A, validated, and B, you have to have method suitability. So let's take a look at some of the rapid sterility test methods, because this is another mechanism that the compounder can use to minimize the impact of these shorter use periods. So rapid sterility test methods are simply test methods that provide sterility results in less than 14 days. There's multiple technologies available. We're going to take a look at a couple of the most common of these technologies that are out. Rapid sterility test methods are accepted by the FDA, but it's on a case-by-case basis. What I mean by that is that it really hinges on that validation that's required. So without appropriate validation, rapid sterility test methods would not be acceptable, but with appropriate validation, they are. So a lot hinges on that validation. You do want to check with your state board of pharmacy. I know that last I heard, the state of California was still not accepting any rapid sterility test methods. So check with your board before you decide to move forward with any of those. Do want to note that any rapid sterility test method is not a USP 71 test. So if you're not doing the full 14-day incubations and things, that it's not a USP 71 test. So your certificate of analysis should not reference 71. It may have the name of the instrument or the type of technology that the instrument uses, but it won't reference 71. So some inspectors may not understand the test method reference since it doesn't say 71. They all have gotten pretty used to seeing 71. And if they see something other than that, then they're going to have questions for you about, what type of methodology is it? Is the test method validated? Do you have method suitability? All those types of things. Do want to note that it's still recommended that if you are using a rapid sterility test method, that you still want to sample your batches per tables 2 and 3 in the chapter. So USP has this chapter available that does talk about rapid sterility test methods, and the chapter says that rapid sterility methods are appropriate for compounded sterile preparations because of the short use periods. So USP is recognizing these test methods. There's also a chapter 1223 that talks about how to validate alternative test methods. So USP has a couple of different chapters addressing these test methods, but do note that even in the title here, they've called it a risk-based approach. And so again, and that really goes back to that validation. If the test method's validated, then the risk is reduced, but so much hinges on that validation. This chapter also says that in the event of a dispute, USP 71 will be the referee test. So USP 71 is the tried and true, and still will be the test that will ultimately decide whether a batch is sterile or not, should that come up in a dispute. So with these test methods, we're going to look at a couple of the different technologies. They all have advantages and disadvantages to them. None of them are a perfect system because of the nature of the test. And nature of the test is, trying to detect microorganisms as soon as possible. So the ScanRDI is a popular system that's out there. Its biggest advantage is that it can provide test results in as little as 3 hours. So when you compare a 14-day turnaround time with a 71 versus 3 hours with a ScanRDI, that can greatly increase the amount of time that you would have to be able to dispense batches. But there are disadvantages to the ScanRDI, and that's that the test method is completely different than 71. So for any traditional microbiologists that are used to classical methods of introducing the drug product into growth media and allowing for the organisms to grow, ScanRDI does not use media. There is no growth period, no enrichment period. So it's a completely different technology. The way the system works is that you filter all of the drug product through some filters, and then a stain is applied to the filters, and the stain attaches itself to any microorganisms present. And so because of the way the test is designed, it can only test filterable samples. So anything non-filterable, like a suspension, would not be able to be tested with the ScanRDI system. I mentioned about the staining of the microorganisms. So the final test result is determined by a technician that is looking at an image of the filter on their computer monitor and trying to determine whether something that's showing up on their system there is a microbiological contaminant, or if it is some sort of other particle. So it's a subjective test that's not always perfectly clear as to whether the test results may result in some additional batches having to be tossed because of sterility failures, or vice versa would be a batch that wasn't sterile being reported as sterile. Because of the nature of the test, again, the way it works with filtration and things, it's a destructive test. So identification of any contaminating organisms is not possible, which is really going to hinder on the ability to be able to do a good out-of-spec, sterility out-of-spec investigation, because you can't identify the contaminating organism, so it's hard to track down source of the contamination and things. The other thing is that the ScanRDI is typically going to be at a higher cost than 71. One of the other popular systems, and there's multiple systems out there, but these are two of the more common, is the CELSA system. This system can provide test results in approximately 3 to 5 days. Its methodology is similar to 71. In fact, the first part of it is exactly the same as the 71. The samples the drug product is exposed to or put into media, and then there's a growth period. It's just that the growth period is shortened from the 14 days to the 3 to 5 days, and then the media is tested for the presence of ATP. Okay, so the methodology is closer to 71 because the first part of the test is just like a 71. It can really test all sample types, and because the system tests for the test, the final test result indicates how much ATP was present in the growth media. It's not a subjective test, and it's not a destructive test. Again, the test is performed by testing the growth media, so the incubation could actually continue on and could grow up the sample and be able to conduct a proper out-of-spec sterility investigation should that be necessary. The disadvantage, obviously, is that it's not as fast as the ScanRDI, still significantly faster than the 14-day USP test, and again, the cost would be higher than the 71. Another disadvantage is that the test may be able or can detect ATP that is non-microbiological. So, like I said, none of them are the perfect system, but they certainly have their advantages. So we took a look at a couple of the resources available to compounders to minimize the impact of these shorter BUDs. That's the USP Compound Preparation Monographs and Rapid Sterility Test Methods. Now Kevin will present some other strategies that you can use at your facility for these shorter BUDs.
Perfect. Thanks so much, Brian. As you can see, the kind of picture that we're painting here from seeing the revisions of 797 and what we've presented so far today is that to get these longer beyond use states, additional testing is going to be our future. So really appreciate Brian going through a lot of those different analytical testing methods and some different considerations there. So I wanted to kind of complete our presentation in applying some various strategies that we can use to quote unquote, extend these beyond use dates for compounded sterile preparations and go through a couple examples. So as we look at and set a goal to say, hey, I have this specific compounded sterile preparation and I'm interested in having it have a longer beyond use date. And so we'll call that, in this definition, we'll call that extending the beyond use date. And so your different options are your sterilization method. And so if you aseptically prepare them as compared to terminally sterilized it, you can certainly apply those longer dates, but you'll still be within that beyond use date table that's in 797. Additionally, considering, do I do a sterility test? Yes or no? And having some abilities to have longer dates there. And lastly, considering the storage condition. Do I keep this at room temperature, or perhaps I need to find a solution for this to be able to provide this at a refrigeration temperature? Perhaps there's maybe units in the hospital that don't have refrigeration that maybe with your strategy you need to consider adding a refrigerator to that unit, or certainly the frozen areas as well. So I went ahead and took that same concept and reorganized the USP 797 table, so you can see that from top to bottom, you're going from shorter to longer dating, and then from left to right, you're going from shorter to longer dating there. And so you can see that, if you're, most are probably using not a sterility test, aseptically preparing, all sterile ingredients. We have our 4 days room temperature and 10 days refrigerated and 45 days frozen. For your particular CSP, that may not be long enough. And so evaluating your options and strategies to extend to a date longer than this becomes really pertinent, and what we'll kind of focus on here. I think it's also pertinent to ask, do I truly need to extend this date? Or what changes can I make to use that 4-day and 10-day refrigeration dating and make that work? And so it's important to look at the pros and cons for whether you don't extend and just, don't do a sterility test, as opposed to extending and using that sterility test. So the pros of not doing it, of course, you don't have any of that analytical testing. As soon as that product is verified by a pharmacist, it can be used for a patient, right? And that becomes really important for quick turnarounds. The cons of it is you potentially could have increased waste. If you have a shortened beyond use date on that product, that could lead to increased waste if it's not being used or is an item that's used infrequently. And then, of course, we have that limited shelf life at room temperature, which may prohibit placement in automated dispensing cabinets throughout the hospital or your practice setting. Now, extending the beyond use dates, the pros would be, of course, we're given now that longer room temperature dating. And so if you pass that sterility test, you have that 30 days at room temperature that you can apply, and that would potentially allow for placement in automated dispensing cabinets in certain areas that may not have access to refrigeration, like your perioperative service space, for example. And since the dates are longer, based off of the usage, you could actually see reduced wastage with that as well. Some cons associated with this, of course, doing that 71 test that Brian outlined takes 14 days. And so you have loss of your beyond use date due to those testing, and we'll talk a little bit more about that. And you also have loss of product. The testing to do that is actually destructive testing, so those samples that you're sending to be tested, you don't get those back. And then, of course, that quarantine delay that we just spoke of. So one of the strategies that we can talk about now is, can you utilize multiple storage conditions to maximize the effective beyond use date of a CSP? So I'll talk through what that means here. So in USP 797, there's a section on storage conditions and talks about using these multiple or different storage conditions before they are used. So such as they are first frozen, and then after some time they're taken out and thawed and then placed in refrigeration and then maybe placed at room temperature. And so the chapter does allow for this and provide some additional information about what would be appropriate. And so it can never exceed obviously the dates that are in 797, and it goes through a couple examples to say that the beyond use dates cannot be additive. So you can't do 45 days refrigerated and then move it to room temperature and add another 30 days to it. You would still have to be within that maximum 45 days, if you will. And so the frequently asked question site on USP that I was discussing further gives some information on this, that you can exceed that original beyond use date, but you can use additional or different storage conditions for the life of that beyond use date for that compounded sterile preparation. So I wanted to represent this visually to show what this would look like. And so we have here your frozen beyond use date at 60 days, refrigeration at 45 days, room temperature at 30 days. I think it's also important to include in here your quarantine period. And so I have a 14-day is how long it takes to do the test, and I always add an additional day. We outsource our sterility testing, so we usually have a shipping day or delay of getting that to them. And so we always add a day, so it's really about a 15-day period for that quarantining process. So when you take that out of the beyond use date, now you have what I deem as your effective beyond use date. So for that frozen beyond use date, you have 60 days, but really the usable beyond use date after releasing from quarantine is only 45 days. So that's kind of the effective beyond use date. So I'll continue to refer to that. And then so you can see for refrigeration, you have 30 days. In room temperature, you only have now 15 days. And so this becomes potentially problematic essentially if you were using the old 797 and now are having to make some considerable changes to your practice. So let's talk about what multiple storage conditions looks like. So in this first example, we're going to be placing this CSP in the frozen state during the quarantining process for that 15 days. And then once that sterility test, USP 71, is passed, we'll take that out of the frozen conditions, thaw that, and have that at refrigeration. And now we have the effective beyond use date in a refrigerated state of 45 days and can be labeled as such. So we now have minimized or eliminated that loss of beyond use date from that quarantining period by using that multiple storage conditions. Another example would be taking an item that is first refrigerated and then moving that to room temperature storage. And then so you can see here that you would refrigerate during the quarantining process for that 15 days, move it to the room temperature storage, and then you would have the effective beyond use date now of that full 30 days at room temperature. And so I want to walk through a case example of doing this with a Category 2 CSP, performing the USP 71 sterility test, aseptically prepared, just using sterile to sterile ingredients, and go through each step to kind of put this together to see what this could look like and how you would go about it. And I use fentanyl because obviously there's additional considerations for how do we keep this controlled and secure during the whole process of this quarantining all the way to dispensing. So you can see we use that previous example. We're going to start with refrigeration for that 15 days while we're waiting for the USP 71 results. And right at the 15-day, we pass the test, and on the 16th day, we'll move that out into room temperature storage, and we'll have a full 30 days of room temperature for that product. Now, it is important that we do have to look at the stability study that we used for that dating, and we have to ensure that it supports both of those storage conditions. So you can see here in this example, we go back to this stability indicating method study, and we can see that they did test it at two different temperatures at 22 degrees Celsius and 5 degrees Celsius, and you can see there that it's within the acceptable assay range, even all the way out to day 93. So both room temperature and refrigeration is checked and supported by the stability study. So we'll go ahead and compound the drugs on the either the manual method or using a robot or other types of automation, and you can see that we end up with our final product here. We have our fentanyl infusion, and the first step we'll need to do is obviously quarantine those products, and so placing those into a quarantining tote and keeping those locked and secured. Brian talked about you have the option to release those prior to getting those testing results back. But really there's additional risk and liability associated with that. So best practice would be to not release those products unless absolutely necessary until you have those passing results. And this is meanwhile while the USP 71 sterility test results are being sent out and the products are being actively tested. Next, we would actually use a secure storage that will, you can see this cart here that is lockable with padlocks. We'll place those quarantining totes inside the padlock, and we'll utilize a walk-in refrigerator to go ahead and wheel these items right into the refrigerator where they'll stay for 15 days while we're waiting for those USP 71 sterility test results. So we get the results back, and it says that it has passed the test. And so now we're in a process, we'll remove those from quarantine, and we'll place those in our dispensing environment, which is our controlled substance safe, which is at room temperature. And then those can be then distributed out to our automated dispensing cabinets throughout the hospital that they're being used, which is room temperature. And those will be out there for the 30 days before those will be expired. So that's really one strategy that you could consider is how do we maximize the effective beyond use date of this product and still staying within the context of USP 797 and within the Table 11 beyond use date table. Another consideration, especially for the compounders that have been doing this for quite some time, they probably have some cadence associated with how frequently they're making batches of these products. So potentially you have a product now that you're assigning, let's say, a 90-day beyond use date. Perhaps you're making it every 4 weeks or every month, and you've been, just really accustomed to that cadence. And every time it comes up, hey, I need to make 600 of these, and we'll send so many out for sterility testing, and you're just used to that cadence. Well, with the new 797, that is going to require some changes, right? And so what this could look like, if we wanted to reduce this down to that 45-day refrigeration temperature that we discussed throughout the previous slides, we'll suggest here a cadence of compounding these every 2 weeks, so making it more frequently. Now, there's pros and cons to doing this approach. One of the pros is you're making it more frequently, and you can actually react to and control the compounded volumes that you're doing on each one of these aspects. So if you're looking at utilization and say, hey, over the past 2 weeks, our usage has really gone down, I may not need 300 for this next time period. I may be able to back that down to 280 or whatever that is based off of the estimates. And so therefore, from a lean principle perspective of doing things more just in time as opposed to in big batches and longer periods of time, we can control that and even reduce waste. Now, it's going to require a lot more analysis and a lot more tracking of the data from the pharmacy perspective, but certainly give some insight to that. The downside of doing this, obviously, is you're going to have a lot more batches. So we just now have doubled the amount of batches, and that means double the sterility testing. So I think that's going to be an inevitable approach here with this new 797 is that we're going to have to be doing more testing. So really the take-home points that Brian and I want you to take home is one, is there's been considerable changes that have been made to the beyond use date assignment in the revised 797 requiring practice changes, right? And so I want to emphasize and underline practice changes is that we can't continue to do these extended and these large batches that we were doing in the previous date under the 797. And so we really need to evaluate our practices and change it to ensure that we're within these chapters or that we are using appropriate methods to extend, such as using those compounded monographs, which leads us to our second point. So those beyond use dates in the compounded monograph can be used if they're being followed exactly and all those additional tests are being performed. The third point is rapid sterility testing methods can be an alternative to USP 71, and Brian outlined some pros and cons to each of those to maximize that effective beyond use date of the CSPs, to really reduce that time period that we're just waiting and having wasted beyond use date as we wait for those testing results. So we can really minimize that and get these products to patients much quicker. And last, multiple storage condition strategies can be utilized to maximize the effective beyond use dates for compounded sterile preparations. So with that, I want to thank everyone for their time and attention for this presentation.
Hi, and thanks, Brian and Kevin. This was excellent. And we've received lots of questions. We'll only be able to answer a few due to time, but I would like to let everyone know that we will prepare a follow-up to answer those questions that we've been asked, a follow-up FAQ document that will be posted online, as well as will go out to those of you directly that are participating in the webinar today. So with that, we'll go right into some questions here. This one is for Kevin, and I'm just going to go ahead and open the questions up here. So Kevin, what BUD storage conditions do you place on the label when you have multiple storage conditions?
Absolutely, and there's a few different strategies that can be provided for certain. So since it's not in the dispensing environment, you can consider labeling it based off of what that final storage condition is. So for example, you could place, if we are intending to use these in our dispensing environment from a room temperature perspective, they can be labeled as such. You can consider perhaps not doing the final labeling until the time of releasing them from quarantine. And so there, I think you have a few different options to consider. It would really just depend on your operations and where that would be applicable. Certainly, I would not label them with both refrigeration and room temperature. I think that can often be confusing. I know that if you look at some conventionally manufactured products, there is some verbiage to say, hey, these are stored under refrigeration, and then upon removal from storage can be stored for X number of days. And so I would say that you really have three different options to consider. I'm sure there's pros and cons of each of those, but that would be up to really the compounder to decide which to select.
Thanks, Kevin. And Brian, I have a question for you related to the USP monographs. Are the monographs manufacturer specific? Example would be Baxter versus Hospira source of a drug or diluent.
Yeah, good question. So the answer is sometimes. Typically, if the monograph is based on using an API, a powder, then the source of the API is not manufacturer specific. But if it's starting with a commercial product, more of a sterile transfer, then those would be formulation specific. But be sure to reference the monograph and see what's there, because looks like the question also was asking about diluents. Sometimes there are manufacturer-specific diluents mentioned in the monograph. So again, be sure to go through each and every detail of the monograph to make sure you're following it as written.
Thanks, Brian. And Kevin, we have another one for you. How can we be assured of stability of a compounded sterile product when frozen?
Absolutely. So one really specific thing that happens in the frozen state, I think there's two aspects I want to bring up. So one is to ensure that your stability test supports that storage condition. So you'll see that a lot of the different stability tests may have only looked at either the room temperature or refrigerated conditions. So you'll have to make sure that you have support for that. Because certain types of admixtures, for example, can crack, like emulsions and so on and so forth. So really important to look at the chemical stability and if that's supported. The second aspect is actually not related to the mixture, but the container closure. So thinking about how it can actually brittle plastic, you can have expanding of the fluid that's in there and cause cracking. So performing a container closure integrity study in that frozen state, I think becomes really pertinent. I think additionally that if that's an option that is really being considered, I think doing the visual verification for leaks upon that thawing and placing out there for use in patients really becomes that much more important to look and to identify, are there any cracks or any leaks or any damage to those container enclosures. So certainly using the frozen conditions, there's some more details and more steps that would need to be looked into to ensure that container closure is intact.
I think one thing I'd add to that also may be that more particles might be generated for a drug product that has gone frozen, because then you have some solubility concerns there too. So as Kevin mentioned, doing the testing at the storage conditions is the best way to ensure that the product's stable.
Well, again, I'd like to thank both Brian and Kevin for an excellent presentation. I know this has been quite a hot topic lately, especially with the 797 revisions going into effect in December. So we do appreciate your expertise and sharing of that knowledge. And we did receive a lot of questions, I think almost 30. So we will be answering each of those questions in an FAQ document going out to attendees, and we'll be posting those on the web platform. And I also would like to add that this presentation will be available for on-demand viewing. So if you'd like to review anything again that the speakers may have said, or if you feel that you know some colleagues that might be interested in viewing this material or in this continuing education, feel free to forward the webcast link out to those folks and invite them to participate in this presentation. And please hang on, because you will be automatically routed to a survey that you need to complete for continuing education credit. And we will also make sure to send that out with the FAQs just in case you are unable to complete that now for any reason at all. And again, thank you today for your presentation or for your participation, and thanks to Kevin and Brian again for their presentations. We really appreciate it.